Hello, I'm Srija. I'm going to talk about exploration of potential molecular players associated with environmental pollutants in the development and progression of upper airway cancers. Particulate matter is classified based on its aerodynamic diameter, where PM2.5 particles are less than 2.5 micrometers and PM10 particles are less than 10 micrometers. PM2.5 particles get inhaled into as far as alveoli and then translocates into blood, whereas PM10 particles reside on the upper respiratory tract, hereby eliciting reactive oxygen species, inflammation, due to which defects in DNA repair, DNA adducts, gene mutation, methylation, and non-coding RNA changes can be seen. Carcinogenesis occurs in two steps, that is, initiation and progression. According to the latest TracerX study by Dr. Charles Wanton and team, particulate matter do not directly initiate cancer, but increase the frequency of pre-existing mutations through genetic and epigenetic aberrations. This is proved in the case of lung adenocarcinoma by the TracerX study, where 3 years of persistent exposure to PM2.5 increased the risk of EGFR-driven LUAD. However, the regulatory players involved in the progression of esophageal squamous cell carcinoma, also abbreviated as ASCA, laryngeal and oral tumors are yet to be identified. We have used gene interaction networks and protein-protein interaction networks to identify the significant disease-associated genes. Protein-protein interaction networks, also known as PPINs, recapitulate genome-wide data. However, they only focus on the proximity metrics, and hence heterodomains are necessary, which can be inculcated using the graph neural networks, also known as GNN. We've obtained the ontological data from Human Protein Atlas, where subcellular location, disease involvement, biological process, molecular function, and protein class were one-hot encoded to reduce the dimensionality of the sparse matrix. And we have obtained the mutational frequency from the GDC portal, where the gene ontological features together with the copy number variations, that is, gains and losses, are concatenated to get the node features. The ensemble gene identifiers were mapped with the ensemble protein identifiers from string to get the esophageal squamous cell carcinoma specific and tongue specific gene interaction network as shown here. Node2vec is a graph representation based model to learn the neighborhood based network features. We have used the gene dragons uh, model to obtain and process this graph neural network. We've used graph convolutional networks, simple graph convolutional network, cluster GCN, and multi-layer perceptron as graph architecture layers. We've obtained the top decile genes from the best model and analyzed using gene ontology and tech pathway analysis. We've mapped these significant genes with the comparative toxicogenomics database to obtain which genes are associated to air pollutants and how are they dysregulated exactly. We've used baseline models such as random forest, support vector machine, and KNN to see if node-only features, that is, HPA and GDC features, or node plus network features, or network-only features, as shown in the figure 3, are performing better. As shown in the figure 2, we have found out that node plus network features were doing good across all the three models, and we also can see from the figure 4 that the gene interaction network follows the power law, where fewer nodes have more connections. Summary statistics show one richly connected component for the esophageal squamous cell carcinoma, and with the diameter of the network, we came to a conclusion that we have to at most use 3 hidden layers in the graph neural network for esophageal squamous cell carcinoma. The results for the tongue cancer were also similar. Out of all the four graph-based models, GCN performed better. So as shown in the figure 5 and 6, we can see that the figure 5 had a better ROC curve, which is of the GCN, than the ROC curves seen in figure 6. As shown by the area under curve values and accuracy along with the F1 scores, we can see that the GCN had a better training metrics on the ESEA-GAN, and the results were similar in the tongue cancer. The p-value of the GCN is also outstandingly significant, as we can see here. As shown in these two figures, top 10% genes of the ESEA are taking a part in the cancer pathways, and we can also see that there are 6 genes that are taking a part in the chemical carcinogenesis via ROS pathway, we hypothesized that few of these 6 genes drive esophageal squamous cell carcinoma and respectively for the tongue cancer, and these genes might be a reason for the progression of the respective cancers due to the exposure of air pollutant. Since we did not have gene ontological features for laryngeal cancer from Human Protein Atlas, we analyzed the differentially expressed genes. We obtained the RNA-seq counts for the esophageal squamous cell carcinoma from the TCGA-ECA project, and we've obtained the RNA-seq counts of larynx and tongue from the HNSE project of TCGA. We've used DSEC2 in R with an absolute log fold change of greater than 1 and p-value of 0.05 to obtain the differentially expressed genes as shown in this table. Using HIPI, protein-protein interactions were obtained. Respective protein-protein interaction networks were constructed using Cytoscape. These proteins were mapped to comparative toxicogenomics database to obtain a subnetwork of air pollutants associated merged protein-protein interaction network for all the 3 cancers. Significant graph neural network predicted top 12.3% ESCA genes were selected in this protein-protein interaction network, and a first neighbor network was constructed to identify any novel disease air pollutant associated genes are immediately interacting with the known biomarkers. From the mentioned DEGs, here the figure B represents the number of DEGs obtained from the shown 5 tissues, and figure A shows its subset of air pollutant associated genes, and we can see that 3 DEGs are common across all these 5 tissues, which are also associated with the air pollutants. This is the first neighbor network of top 12.3% GNN predicted ESCA DEGs. The yellow nodes are common among all 3 cancers. In the presence of increased ozone or particulate matter, we see a decrease in the TOX2 expression. TOX2 and AKT3 are both proteins involved in cellular signaling pathway. TOX2 might influence the expression of genes regulated by AKT3. We also see increased expression of DPF1 in presence of increased ozone or particulate matter. We also hypothesized that OBSC1 regulated by FGFR1 can be strongly associated with the air pollutant driven upper airway cancer, where FGFR1 drives the tumor growth by activating the downstream signaling pathways involved in cell proliferation and survival.